Hello and welcome to this video presentation on DNA biosensors. This video was produced as an educational segment for Louisiana Tech's Nanotechnology education series. In this video, you will be introduced to DNA biosensors, their mechanisms, and their uses. Some of the most common uses of DNA biosensors is the early detection of debilitating and morbid diseases such as AIDS, cancer, and influenza. AIDS is responsible for 25 million deaths till date from its date of first detection in 1981. AIDS is the result of untreated HIV infection. This can be prevented as a disease in HIV infected patients by early detection and treatment. AIDS patients do not only die of AIDS, they also die of social stigma. In Papua New Guinea, AIDS patients are buried alive to protect other family members from acquiring this disease. Cancer is another such disease we know that causes high mortality. If cancer were detected early, then metastasis could be avoided and recurrence could also be avoided. Influenza is a pandemic declared as a pandemic in 2009 by the WHO. The swine flu pandemic was the worst we know till date. Influenza has the properties of changing or adapting to treatments very quickly. One influenza vaccine administered 1 year is useless the next. Influenza also appears as symptoms in human beings quite late. The basic symptoms like fever, cough and cold are dismissed. In this video, we will discuss DNA biosensors, what they're made of, and how they can be applied in treating or at least detecting such diseases.
So what are biosensors? A biosensor is a self-contained integrated device capable of providing specific quantitative or semi-quantitative analytical information using a biological recognition element capable of indirect spatial contact with a transduction element. So in simple terms, a biosensor consists of a biomolecule attached to a physical device called transducer, which is connected to a detector, and this transducer transmits the signal of a biomolecule recognition event, and this is then detected using a detector and observed by us. Some of natural biosensors that we know of really well: our nose, skin, ear, eyes, tongue, and even the canary bird has been used by miners in coal mines to detect poisonous gases. So the first biosensor was a blood oxygen level detector made by Leland C. Clark at the Ohio University, and this is useful for detecting blood oxygen levels in diabetic patients. Next, we look at the biomolecular probes that are used in biosensors: DNA, RNA, PNA, aptamers, DNA zyme. These are all various forms of DNA. Apart from these, enzymes, proteins, and antibodies are also used as in biosensors. So let's look at DNA biosensors. In DNA biosensors, we use DNA, RNA, PNA, aptamers, or DNA zyme as the probe. Let's look at the DNA structure now. The DNA structure consists of two strands of DNA which are joined in double helix form. The two helices are joined by four base pairs called adenine, thymine, cytosine, and guanine. The adenine binds only with thymine and cytosine binds only with guanine, and these are bound by hydrogen bonds. And the backbone of the two strands consists of sugar phosphate molecules. And these are hydrogen bonds in the adenine, thymine, and hydrogen bonds in guanine and cytosine. So the working principle of a DNA biosensor is DNA hybridization. A single-stranded DNA is first attached to a substrate, and this is then brought in contact with the analyte solution containing the DNA to be detected. If the probe DNA and the analyte DNA have the same base pairs, then the hybridization signal that we get is very strong. Here we see a complete matching of base pairs. Even if one of these is not matching, then the hybridization signal will be weak. Here we see that the probe DNA is attached to a fluorescent molecule, and this is then used to detect DNA hybridization. There's a change in the fluorescent signal after hybridization with the target. So the DNA biosensors that will be used for diagnosis will use a probe attached to some kind of molecule for detection, and the sample and the solution containing the target DNA. The solution will have large number of various DNA with different base pairs. So if there's a matching of base pairs, then you'll get a strong hybridization signal. And based on this, we can detect the DNA molecules associated with various diseases like AIDS and cancer. So how do you attach a DNA molecule to a substrate? We can use covalent bonding, electrostatic adsorption, or streptavidin-biotin interaction, and they can use any of these methods for attaching DNA to a substrate. Next are detection techniques. There are 3 detection techniques available. First are electrical. In this, a change in current or voltage due to hybridization is measured using amperometric, potentiometric, or electron impedance spectroscopy. And in this case, there are some researchers prefer to use another molecule that may increase the hybridization signal on binding with a single-stranded or double-stranded DNA based on its affinity. Then in the optical method, change in fluorescent intensity of dyes and change in refractive index due to in-surface plasmon resonance and Raman scattering and chemiluminescence are used for detecting DNA hybridization. In piezoelectric based methods, a quartz crystal microbalance may be used to detect change in resonance frequency or mass or surface stress due to hybridization, or a microcantilever that is sensitive to small changes in mass can be used to detect DNA hybridization. In all these techniques, the researchers have demonstrated that even if you have as small as nanogram, that is 1/1,000,000th of a gram of the DNA of the particular disease, it can be detected with great sensitivity and in a very short time, like few minutes. And next we'll show how DNA hybridization occurs based on this tutorial.
Often a particular piece of DNA or RNA of interest needs to be detected in a mixture containing many pieces with different sequences. One way to do this is by hybridization. Hybridization refers to the process in which 2 complementary pieces of DNA, or a piece of DNA and a piece of RNA, anneal to form a double-stranded molecule. The piece of DNA or RNA used to detect complementary sequences is often called a probe. If the probe is labeled, for example by making it radioactive or fluorescent, then any pieces of DNA or RNA that it hybridizes to will also be labeled and can be detected. Before it can be used for hybridization, a double-stranded DNA molecule must be separated into single-stranded DNA molecules, or denatured.
And there's been a lot of improvement in DNA biosensing recently based on which depends on the use of nanomaterials. Some of the improvements that have been observed is increased sensitivity and detection of multiple analytes. Actually, the sample that blood sample or sample of gene that you obtain from a patient will have different types of DNA with different base pairs. So, by attaching different single-stranded probe DNAs to a surface, multiple DNA molecules can be measured, and you can detect more than diagnose more than one type of disease at the same time. Nanomaterials provide a large surface area, so this can be used to attach a large number of probes to the nanomaterial, and this can even a small device can be used to diagnose different diseases. And device miniaturization is another important advantage, although the cost may increase due to the use of nanomaterials. And this picture shows a microarray that has been used to detect tumors, and a fluorescent dye has been used to detect the hybridization signal. And each of these in each of these holes there's a different kind of gene, different kind of DNA, and based on the matching of base pairs, you'll get a different type of signal for in each case.
We next discuss the various applications of biosensors. Biosensors can be used in analysis of food after processing, drug development and its effects on human beings, forensic studies, studies of biomolecules and their interactions, industrial process control, and in the diagnosis of gene sequence related to diseases such as HIV, cancer, and influenza. DNA biosensors can also be used in environmental monitoring techniques. Companies that currently manufacture biosensors are Abbott Point of Care Incorporated, Affinity Sensors, Neosensor Limited, Siemens Healthcare Diagnostics Incorporated, and Animas Corporation. There are many commercially available biosensor devices, some of which we'll show here. One of them is the pregnancy testing kit. The other one is the glucose monitoring devices for diabetes patients. The third one is a device that detects respiratory diseases in human beings. The major advantage of using these devices over current or previously existing devices is that they're terribly accurate, they're very precise, and they're extremely fast in detection. Sensors used by man. We come to this really important question. Do we want to invest time, money, effort into making and perfecting biosensors and applying them to human disease detection, or we want to simply sit back